Okay, I think I can figure this out. I don't know if it's like walking and chewing gum and pointing. I talk a lot, but I don't necessarily talk in front of a group a lot, so bear with me. So as Jean said, Simon and I founded Hope for Annabelle 6 years ago now. Um, and we have been part of the HC world for that many years. To us, it's kind of flown by, and I think we have nothing but tremendous respect for all of the families and parents who have been here for so many years before us. So we have been working together as a community to kind of support research together in a careful, thoughtful, and very strategic way. And so what I want to talk to you today about is how we approach it, how we have kind of formulated a strategy around research that is a guiding principle for all the foundations together so that we can really make the most of the very limited resources that we have. So the first part of my talk is going to be discussing that strategy framework, the objectives that we have as a community. And then I will be going through an update of the different projects that we're currently funding. So first of all, when we were thinking about what our mission was as a community, I think we kind of iterated a few times on this. I know Natalia, who's one of my main partners in crime and who will be talking to me, who will be talking next, doesn't necessarily agree with this mission. The word that she has issue with is the right treatments. And she says, how do we understand, how does that convey anything? And I think that's in some ways my point, is that we are trying to figure out what the right treatments to fund and what the right treatments to pursue should be. And so that is part of our mission. But the first piece of this really, though, does kind of rest on patients, understanding the needs of the patient population and trying to meet all of those needs, working with time constraints and also working with funding constraints. So from our mission, we get to 4 different objectives that are all kind of founded in this interest in what our mission needs to be. So addressing the patient population comprehensively. We have a large population and it's variable, and we need to make sure that we are doing what we can in supporting research that benefits all. Advancing research that has a therapy focus. I think we're very interested, and we've had a lot of success in basic science, but we have also been very interested in how that translates to therapeutics development. We are also working as quickly as we can, but as carefully as we can at the same time, and we're balancing those two constraints as we move forward. And the final piece is funding deliberately and carefully and maximizing the limited dollars that we do have. So the first piece here is the patient variables. We have a really variable patient population, and they have variable requirements, and it is our mission and our objective to address all of these requirements so that we aren't leaving any patients behind. I have a chart here of different factors, different symptoms, different preferences, but this is kind of a way to put a methodology on the kind of suffering of our patients. I mean, that's the kind of crux of what drives us is addressing that suffering, addressing the symptoms that our patients are experiencing every day, and the kind of hardships that their families are also facing. So that's kind of the crux of it. But as we are moving forward, we really need to be sure that we are, addressing all of these different variables. And there may be others that, this is very much a working document that we are most happy to incorporate extra points into. So for instance, Kathy Swoboda might have other points where, we need to address this piece of the patient population. And this is kind of a work in progress. So with the, variable patient populations, I think it has become clear to us that what we need to do is support a portfolio of projects. Looking at a single therapeutic strategy just isn't enough, and there are a number of reasons for that. So different therapies may be more effective or less effective for different patients, depending on the variables of the patients themselves. So that's the first fact, that we need to kind of spread resources so that the most patients possible can benefit from them. The second point, of course, is that different therapies can and should be layered on top of each other. A lot of other rare diseases have benefited tremendously from that sort of strategy. And then third, we don't like to talk about the risk of failure, but that is absolutely the case with all of the kind of new development research that we are pursuing. We are very much, on the cutting edge of various different technologies. And failure is certainly a possibility. Even if we vet projects very carefully and approach them very carefully, that is still always a possibility. So we are pursuing deliberately multiple therapies in multiple paths as we move forward. So this portfolio is kind of spread across these 4 different focus areas, and we're using 4 different approaches. We're funding 4 different focus areas, I guess is what I'm calling them on this slide, of where we are supporting research. So the first, and I'll go through these in greater detail once I go through the different projects and where we are on each. But the first category is correcting the genetic defect directly. The second is reducing symptoms by having an indirect kind of a target. And those are completely therapeutic development centered. Like that is where we are with those. But we're pursuing all of these things in parallel, and we are not, and I think that's an important point, because as we are pursuing these therapeutic development efforts, we are also kind of looking to see what we can learn from basic science and how we can apply the findings from basic science into new projects going forward. And I know we've kind of learned about mechanisms in AHC as we're working on projects that are maybe designed to be targeting a therapy, but yet we're still kind of finding out more about basic science at the same time. So there's a real bidirectional relationship between what we're learning in basic science and what we're learning in projects that have a therapeutic target. And at the same time, in parallel, we are also trying to build a research infrastructure that makes research easier for scientists. We want to be an easy target as a disease as much as possible. We want to prepare early for clinical trials and support clinical trial readiness early, so that we can kind of get all these pieces in place that we will need down the road if there's a therapy that proves to be effective and we can move it forward. Sorry, my daughter's in the front row playing with my purse. So if there's any commotion, or if I start giggling, it's because she's making faces at me. So sorry about that. So the next piece is working kind of quickly but carefully, and moving research forward at a good pace, which is, it's a balancing act. We all have patients that we love that are getting older, and the therapeutic window might be closing for some of them, and we're acutely aware of that. We're also very much aware of the fact that we need to kind of proceed cautiously and carefully. So there are a number of ways that we try to implement that strategy. We try to make sure that projects, when we are funding them, that the scope is streamlined, that the kind of questions that we're asking are targeted, and the outcomes are kind of predicted. We've also been able to collaborate in such a wonderful way with so many wonderful scientists. And I think we have been so lucky to be able to kind of leverage findings in real time and kind of translate them into new projects as we're going. We're not waiting for publications years down the road. We are, finding out something the same week that something happens in a lab, and we're kind of able to either maybe pull the plug if things aren't going well, or redirecting our focus into new areas of opportunity. And then finally, the preparing early for clinical trials is another way that hopefully we can kind of move things forward quickly for the disease. So the fourth kind of pillar of our strategy is funding projects carefully, and making sure that we are really deploying our funds that are so difficult to raise and are so precious to the community, that we're deploying them in a way that we should. So the first kind of piece of this is picking the best of the best, working with the best scientists possible, weighing projects and different kind of areas of therapeutic development against others. We kind of have to weigh things in a very relative way so that we can kind of figure out where, in relative terms, our priorities need to be. As I said earlier, we also have to focus on things that are very that have a direct line of sight to a therapy coming out of the research, whether it might not be direct, but it needs to at least kind of anticipate where that's going to fall. The other really important part too is that we are able to invest in early stage research in a lot of different paths at the same time, because this is kind of the cheap part of research. If we are, if we do have a project that becomes successful down the road, and say it's a genetic therapy of some kind, the costs for synthesis, the costs for toxicology, those costs just increase exponentially. So this early stage is where we really can diversify our efforts across a whole portfolio of different approaches that might prove fruitful, whether for multiple patients or for specific patients. And so that's something that we're certainly very interested in pursuing. And finally, if there are shortcuts we can take in leveraging adjacent findings in different fields, this is something that we're certainly very interested in, whether it's looking for biomarkers that have been approved by the FDA and other clinical trials that, we could do a very targeted hypothesis-based screen in our patient population. Those are the kinds of places where we might be able to kind of cut costs and cut timeline there. But I think that the main kind of hypothesis of what we are going with for our strategy is, multiple approaches need to be pursued in parallel. And that is kind of the best way to increase our chances of success. So finally, the key piece of all of this that really makes all of this possible is collaboration with the scientists and researchers that we work with. That has been absolutely, I think, critical to the effort that we've been putting in the last few years. And it's something that's been really successful for us so far. So I made this graphic. Simon and I talked about this graphic on the right. And I, put it into the PowerPoint. And when I made it, I was really envisioning the kind of blue boxes as being a patient function rather than a researcher function, which is, perhaps a divergence from, what is standard. But I really think that as a community, we've done a really good job being involved in developing, helping to design. Some of you may disagree with me on this, but I'm looking at you, Al. But we have tried to kind of insert ourselves into the process where we can be, as equal partners as possible. And I really do think that there is a great deal that sophisticated research groups like ours can hopefully bring to the table in terms of bringing teams together, fostering collaboration, raising money, centralizing information and data. There, I think, are a lot of ways that, patient-led efforts can be really, really game-changing. We've got tremendous scientists that we work with, but hopefully with kind of our added input as well, that we can all work together and kind of move things forward as well. So that is that. So that's kind of where we are strategy-wise. It's kind of the premise that guides all of our research that we have been supporting, that we are currently developing. This is how we think about things. And we thought it was very important to present that thought process to you, because it's something that has been developed over many years. It hasn't been articulated and put on paper and presented to the patient groups. But I think that is something that's, understanding where we're coming from and hopefully where we're going, I think is really important to share among patient families. So that's the first part of my talk. I'm happy to entertain any questions about this. And then the next part of my talk will be about the different projects in kind of a less technical sense than Al presented this morning, with, the basics of what the projects are, and then some updates on status updates that are right now. So if there are any questions on, strategy or how we think about things and approach things, I will open that up right now, and then we can move on to the next part.
You were sitting closer, Kathy, before.
Oh, sorry, I snuck in the back. I just wanted to attest to how refreshing this is, because I've sat on many different, rare disease groups and so on. And I mean, not all scientists come to this easily, but I got to witness a great interaction with a recently funded project, and where the investigator put in the proposal, a very senior investigator who's very well respected in the field, you guys very tactfully and carefully and appropriately gave critiques and cutoff points where you want to say, okay, that sounds great, but we want to focus on this part of it and rewrite it. We've done this with the SMA community too. And it actually is not always well received because I think the scientists and people putting in, they put all their whole heart and soul into this grant writing, and they are very knowledgeable in their fields and so on. But I want to say how helpful it is to manage your resources wisely, because you are a small disease, so a rare disease organization. So I've been very impressed by that.
Any other questions?
Super. Okay, so let's move back into this slide to kind of give a framework of the different projects that we're going to go through. And we're going to start with the correct, the defect, that kind of category of projects that we're funding. So there are 3 different projects here. They're based on 3 different ways to correct the defect. And to kind of oversimplify, one's an editing strategy, one is an adding or overcompensating strategy, and one is a silencing strategy. So that's kind of, I'm going to try to present them in the most kind of layman terms possible so that we can make sure that that we're all on the same page here. I think Dr. George did the hard job this morning of getting into the details. So this will be much less detailed. So the first project that we have ongoing is the gene editing project with David Liu's team from the Broad. So this is the editing approach where the Liu lab is creating machinery to correct the genetic defect that's present in AHC. It's a permanent fix. It would be a one-time treatment. It would be mutation specific, and they are working on 4 different ATP1A3 mutations currently. One of the questions with this therapeutic strategy, as with some of the other kind of genetically focused ones, is what the therapeutic window might be. They are investigating that currently in the project. So as far as where the project is, we've been underway for about 18 months. We just won an award from CZI that was awarded, I can't remember when it was officially awarded, but it was in the fall sometime. We won the first $1 million already, and there is a second $1 million that is available after 2 years. It's a non-competitive award. So we're somewhat optimistic that if things go well, that we can kind of put down the $2 million number instead of the $1 million number. But we're very, very pleased not only to have the grant, but to be working with David Liu, who is just absolutely a leader in his field. I mean, if we're talking about funding the best of the best, I think that's very much the category that David falls into, which is absolutely fantastic. He has had success elsewhere. I think his work in progeria has been very well publicized. That's a disease that, you know, we might be familiar with from the movie I Am Sam, which is such a tearjerker. But it's just amazing to have him on our team. So they have been working first on a strategy for mice, and they're about to begin testing in mice. The first round of testing in mice will just be a DNA analysis to look for editing efficacy, and then behavioral tests in the mice will follow. And then we are also starting with Dr. George at Northwestern. We will be looking at primary patient cells, iPS cells, and Dr. George's lab. First iPS cells and then neurons, I think, is the plan. So that's also really exciting. Those are actually, you know, patient lines. So we'll be getting some really interesting results, both in vivo and in vitro, which will be super. I think the other thing to add here is that, you know, I think the Liu lab is actually really excited about the results that they're seeing for AHC. I think the editing efficacy is really kind of near best case levels. So they are seeing kind of the optimal kind of end of editing and very minimal deleterious byproducts, which is absolutely super to have them excited about this. So that is the CZI project for gene editing. So the AAV mediated gene therapy project has been ongoing for a number of years. This is something that we're probably all familiar with for a long time. But the mechanism of this is adding additional functional copies of ATP1A3 in order to kind of overcompensate or compensate for the mutant proteins that interfere with pump function. So the the mutation in an AHC patient's ATP1A3, if it's based on an ATP1A3 mutation, of course, creates issues with the pump, as Al discussed this morning. And so we are trying to minimize the extent of damage by adding additional functional copies to kind of dilute the effect that the mutant proteins are having on the pump. So again, this is similar to gene editing. It would be a one-time treatment. We are trying to determine the optimal route of administration. In mice, it's going to be injections that would be more invasive for patients. So there's been a lot of work done to kind of see where that's going to be in the future. So the other thing to mention here too is that while we are testing this on D801 mice, the vector that we are using is something that is mutation agnostic. So any patient with an ATP1A3 mutation could benefit from the vector. And again, similarly, as a genetic therapy, we're not sure what the therapeutic window is, but that's something that we'll be looking at. So we had some issues with this on a couple pilots. Does this sound okay? We've gone through Sound okay? Okay. We've gone through a couple pilots and we've had some encouraging results, but not kind of resoundingly encouraging. We've had so many mice die that we haven't been able to achieve the kind of statistical significance that we would want to see, particularly if we want to move forward with us. But as we were resolving the issues with the mice, JAX has been rebreeding the mice to create a stronger, more robust mouse model that still maintains the same phenotypes, but just doesn't, die at the drop of a hat. We've also been trying to prove the concept in vitro, which we did with Dr. George earlier this year, or maybe last year. And so we are, the study is ready now to resume. We are going to be starting this summer with injecting mouse pups. We're going to run a number of different behavioral tests on them, and we are hoping for initial data to be back by the end of the year. So the other thing to note on this is that JAX, the Jackson Laboratory in Maine, has been working really closely with us on a number of things. I think we have been fortunate enough to develop a really good relationship with Cat Lutz, and they are using AHC in many ways as a poster child for their rare disease capabilities. And so they are performing the recharacterization work of the D801N mouse, as well as a number of other mouse models for no cost to the foundations. They're also doing this pilot at no cost to the foundations. They've offered up a number of things that have been really, really phenomenal for us. So that's also really great that we can go through another pilot, but not have to pay for it. So the next, now I'm at the silencing strategy of ASOs. So Dr. George has gone through this very in much more detail. I will just kind of reiterate the basics. An AHC patient with a mutation in ATP1A3 has one good gene copy and one bad gene copy. These are called alleles. It has been preliminarily demonstrated that knocking down the bad copy or silencing the bad gene and the resulting mRNA and protein, that has a positive effect on a neuron at least. So we think that the therapeutic strategy is one that really holds water for us. So we are moving forward with that. ASOs can be designed around either the causal mutation or around a patient's specific sequence in DNA. ASOs need a place of variation or divergence from the normal. And so once that can be identified, those are kind of the targets for an ASO. So ASOs ultimately could be designed that would work multiple patients with one mutation in common, or there might be SNPs. There might be other variants in common in their entire. This one's tripping me up. Mary's kind of nodding at me, but it's hard to kind of oversimplify this enough. But ASOs can be designed for a patient, but hopefully an ASO designed for one patient could potentially have benefit for multiple patients. This is a more frequent administration with a spinal tap is typically how it's done. The half-life varies for different ASOs, but typically I think we would be looking at administration every 6 months or so. Again, we're not sure exactly what the therapeutic window is. I think that's very much an AHC specific question. There have been patients treated successfully with ASOs who are 60, but not for AHC. So there's still a number of questions. So we're working on three different areas for ASOs. Mary Saladino has been working tirelessly for Henry to develop an ASO for her son Henry. She has had ASOs designed, and Dr. George is testing them in his lab on Henry's iPSC neurons. So that's kind of the first line of work that we've been supporting. Mary's kind of doing the hard work, and we're trying to support her where we can. We're also looking at whether ASOs are a good therapeutic strategy more broadly with Dr. George and his lab. And we are looking to Dr. George's lab would be looking in vitro to confirm if this was a good therapeutic strategy, but we're also looking to confirm in a mouse model if an ASO designed against the mouse could have a benefit on a mouse, on a living organism. You know, how would an ASO cure a mouse? So we're looking at that as well, and that's another project, incidentally, that JAX is providing at no cost. And we are also looking at other scientists who can support this effort moving forward, including Tim Yu at Boston's, who's really one of the best in the country, another fun, the best of the best kind of situation. Okay, so strategies to reduce symptoms indirectly. We have two projects that are in development here that we are working to kind of get proposals finalized and to get contracts going. But we do have one project that is already underway with Dr. Clapcoat in the UK. And he is looking at using endogenous WABA antagonists to improve pump function. So the idea behind this is inhibiting an inhibitor. So we all, we've heard how the sodium potassium pump is critical to AHC, and we've heard, or is critical to life, and an AHC is impaired. But, AHC mutations and dysfunctional proteins are not the only things that impact pump function. And so we are looking to address another suppressor of pump function. If we can eliminate that suppressor of pump function, the thought is that there might be some therapeutic benefit to kind of limiting the other pressures that are reducing the functionality of the pump. So I think one of the things that's interesting here is that endogenous WABA is triggered or released in response to stress. And we kind of see that in our own kids in a parallel situation, that there are triggers for our kids, often from stress, that lead to symptoms. So it's a kind of interesting and compelling project because it, does have that kind of clinical relevance there. So a treatment from WABA antagonists would be repeated or chronic administration, likely, systemic administration. All ATP1A3 mutations would be likely to benefit, although we're not quite sure if the non-ATP1A3 mutations would also benefit or not. It would be easy to hypothesize that potentially a greater kind of age range of patients might be favorably impacted by this kind of a strategy rather than a genetic strategy, but that is still something that, isn't confirmed. So Dr. Clapcoat is testing this as a therapeutic strategy in mice. He's looking at 4 different compounds. Worth noting about the compounds is that 2 of them have been either approved by FDA or approved for safety, but not efficacy through FDA, which means that they would be easier to repurpose and could be candidates to kind of move through an expedited path through FDA, which would be very nice. So Dr. Clapcoat is subjecting mice to behavioral tests that are very similar to the tests that we're doing at JAX on our mouse models, on our different mouse models there. And he has been also participating on calls with JAX. We meet monthly. Everybody's kind of really happy to be collaborating and working together. And Dr. Klapko's a big part of that too. So the next kind of area that we have been investing in is building a research infrastructure. Natalia is going to talk about all of this animal cellular model business. So I will move to the next slide. So we've been working to promote some clinical trial readiness, aggregation of patient data, looking at biomarkers, looking at things to kind of make future clinical trials possible and more smooth, whether it's whole genome sequencing, which is something that Simon's been really passionate about for rare disease in general, and could be useful in a different and very specific way for AHC as well. And also producing a new natural history study. There have been a number of natural history studies to date. Data from some of them have been very limited. So we are trying to make sure that we have natural history data accessible to the community moving forward. And we're also trying to figure out some good biomarkers that can show either disease variability or progression in AHC that could be used either diagnostically or as a measure of efficacy in a clinical trial. So we also have a number of new projects that are kind of in the pipeline that we've been developing. And they kind of are based on a few different concepts, which is, leveraging the basic science work, thinking about what we know already, the different kinds of models that we have already and how we can use them differently and in a more focused way towards therapy development, leveraging findings in adjacent fields that have, there's been a lot of advances since we started working on the AAV project 5 years ago, for instance. So we need to kind of make sure that we're keeping up with relevant adjacent research. And, pursuing these new projects is feasible and possible because we have been, able to invest moderately in these early stage pilots. So I think we also structure a lot of these new projects as kind of a small bite-sized piece to investigate a concept. We see if there's any, any legs, teeth, whatever the analogy is, and then proceed with more funding in a larger project that way. So we are not tapped out. We are still looking actively for projects. We're still, actively considering where we may have gaps in what we are doing and where we can do better. I think, there's a lot of room for growth, but our time and capacity is somewhat limited to find it. So we are relying, I think, on scientists and researchers to kind of help us identify new therapeutic strategies that could be targets for us. I think that would be, really important going forward because we have the capacity to add more projects, right? We have the ability there are projects in our pipeline. We could yank the chain on those and find better ones, but we need to be looking out for new projects all the time. And this requires a tremendous amount of, work and research. And Natalia, who's talking next, is kind of the big reason that this is all possible. But this is all a work in progress, and it is something that we've been grappling with for years about how we can make a difference in our kids' lives. It's not just about ending AHC, which is, of course, our mission and our objective, but I think equally as important is the idea that we need to make patients' lives better who are already, going through all kinds of torturous, not to overstate, but truly torturous kind of conditions that they have to deal with on a daily basis. So, those two paths of making lives better and ending AHC are what drives us forward. And, it's the first thing that I think about every morning. And usually I'm thinking about it before I go to sleep and I can't sleep because of it. But that's I think how we all have to be is motivated. And I do really think that we are going to kick in the door and claw some eyes out. Maybe that's too far, but we are very determined, to make a real difference. So Thank you for letting me talk. Please ask any questions.
He's going to test you on his study.
Yeah, that was really wonderful. It's really refreshing to see a scaffold. I think that was badly needed. And I think a lot of rare disease foundations call it a roadmap. And I think you actually showed the diversity of efforts in this disease, which reflect the diversity of aspects of this disease. I think you need to probably think about, there's a lot of roadmaps here, and maybe even think about developing roadmaps for each of these different components that really take it out year by year. Because I think, again, I think I'm sure this is going to undergo a lot of evolution, but I would just say, I think it's great to have a scaffold. So the one thing this is just an idea that I haven't heard talked about in this rare disease community, but it's very common in others that have multiple competing studies, research studies, natural history studies and things like that, where it's easy to lose track of patients who are in multiple studies and have biospecimens over here or data over here. There are these, there's something called the CRID. I don't know if anyone's ever heard of that. It's called the Clinical Research Identifier. It's free. Patients or families apply online and you get a number, a unique identifier that then you can share with Rare X or the George Lab or somebody else. It's a de-identified number that represents your child for their biospecimen, for their clinical data. And I think this is the time to start thinking about doing that. And families have to request this and then have to share that. So if there's 5 natural history studies going on, that one individual in all 5 studies would be identifiable. And so I think this is something I'm happy to share that with others. It's free. There's a guy in Ireland who I know who runs it, and it's really widely used in many rare disease communities. So CRID, easy to remember. There wasn't really a question there, by the way. Just in case.
Well, I meant it when I said I'm very open to ideas and suggestions and input. So that's definitely what I mean. You write it down. Oh, right, okay. My secretary is going to write it down.
Oh, yeah.
Thanks a million for that talk. It was really incredible to see it. It actually struck me that it looks like a spin-out company from a university that I sit on lots of these panels that judge it. So have you considered the commercial angle here as well, of tapping into the resource of pharma companies, small companies, but also large companies, and looking at ways how you can engage them, but also tapping into venture capital funds, which are very, very large. And, you could move things along at a much faster pace with more money. Is that something that's on the agenda or something that you're contemplating?
I think a lot of the reason that we did want to put something together that shows our methodology is so that we can make a case for investment to others, and that we can share this more widely and have kind of a compelling argument for people to join our team and get some funding that way. Because I think one of the things that we need to do besides being strategic about research is being really professional about fundraising. And I think that's where kind of our next road bump is. So if hopefully this can kind of help towards addressing that, portraying what we're doing in a careful, thoughtful way. Hopefully will get us towards that goal.
So yeah, thanks, Matthew. I just wanted to jump in on that particular topic too a little bit, because we are part of the thesis here is that we've been trying to get things to proof of concept so that we can de-risk for pharma to jump in and move the ball along with the expensive part of the therapy. But we've also been speaking with very targeted pharmaceutical groups, especially smaller ones, on each component of the project earlier. And we've also been trying to protect IP to the extent possible so that we can make sure that patients are protected, that we can keep it cheap or free for patients to the extent that we can. And where we can't, we can at least guide the path towards clinical trials and beyond.
The other thing I should say, and probably didn't emphasize enough, is that, you know, we've had a lot of success collaborating as a community. And I think that has been some secret sauce in the mix is, you know, that we are not just working kind of in isolation, but that we have a really big team of people. Sorry. I'm not good at public speaking. I don't know that I've done it more than twice in my life, but I think that's, you know, totally lost my train of thought. Collaboration. Yes, sorry. That has been absolutely instrumental to what we're doing. And I think, you know, it's not just the collaboration that we're hoping to foster between the scientists, but it's also this collaboration between the different foundations, whether it's AHCF, Cure HC, for Henry. We've all been kind of working together. And I think we see ourselves as servants of the community rather than, you know, trying to prove anything else. And I think we do feel the weight of responsibility for what we're doing.
Wait, one question.
So how can families help?
Well, one way, Dr. Sharma is here with a natural history study that we're working on. So participating in natural history studies, there she is there. Annabelle, my daughter, saw her today and had a really fun time. So that's one way for families to get involved. You know, as Josh said, we also do, we have more things on our plates than hours in the day to do them. You know, we do stay up till all hours thinking about things, and, you know, Natalia will text me at 3 a.m. sometimes. So we also do kind of need bodies in various places. One place might be kind of monitoring some of the patient forums in Facebook to kind of synthesize information about where patients are suffering. There's a lot of kind of noise there, but I think there's also some really good information about the ways that patients, their symptoms are affecting their lives and could kind of be targets for us in the future when we're trying to kind of design therapies. I mean, what I have put before you is certainly aspirational. I'm not trying to claim that we have you know, solved the issue of addressing all of the patients in our community, because that's a work in progress as well. You know, I don't want to overpromise and underdeliver. So I think it's better to kind of be moderate, but that would be, you know, there are ways for patient families, even without kind of scientific background, to get involved.